Hello everyone, today, we will analyze the bronchial fibroblasts respond to house dust mite challenge with cytokine secretion, role in allergic airway inflammation. This study was collaboratively completed by Professor Wang Xiaozhi's research team from Bingzhou Medical University Hospital and Professor Dong Liang's research team from the First Affiliated Hospital of Shandong First Medical University, and it was published in iMetaMed journal. Fibroblasts regulate the progression and resolution of pulmonary inflammatory responses through various mechanisms and play important roles in many processes. Su et al. Found that stromal cells and myofibroblasts in the outer matrix regulate disease progression by recruiting pro-inflammatory and anti-inflammatory immune cells in allergic airway inflammation. Osei et al. Discovered that interleukin-1 alpha/beta induces the production of granulocyte macrophage colony-stimulating factor and thymic stromal lymphopoietin, while suppressing the generation of collagen, fibronectin, and periostin. Drake et al. Observed that tumor necrosis factor alpha significantly increases the mRNA expression and protein secretion levels of interleukin-6 and eotaxin in pulmonary fibroblasts. However, the response of bronchial fibroblasts to HDM stimulation and its underlying mechanisms remain unclear. The highlight of this study lies in uncovering the early characteristics of HDM-induced bronchial fibroblasts. Extracellular matrix production is inhibited while inflammatory signaling is activated. In patients with allergic asthma, the cytokine CCL2 is highly expressed in airway mesenchymal cells. Bronchial fibroblasts are important airway mesenchymal cells. We stimulated bronchial fibroblasts with HDM and performed gene expression analysis. Using differential criteria, we identified differentially expressed genes, including IL-6, IL-1 TNFSF15, CCL2, CXCL1, CXCL2, CXCL3, CXCL5, and CXCL6. Among these, TNFSF15 and CCL2 are closely associated with eosinophils, while IL-6, IL-1 CXCL1, CXCL2, CXCL3, CXCL5, and CXCL6 are related to neutrophils. In contrast, the expression of fibronectin, COL1A1, vimentin, and TGF-beta1 was downregulated, although their fold changes were all below 1.5. Go enrichment analysis indicated that these differentially expressed genes are closely linked to programmed cell death pathways, NF-kappa B and MAPK inflammatory signaling pathways, as well as neutrophils, Th2 cells, and eosinophils. These findings suggest that early-stage bronchial fibroblasts under HDM stimulation are also involved in the development of asthma-related airway inflammation. Western blot analysis revealed that HDM stimulation did not alter the expression of vimentin or alpha-SMA in bronchial fibroblasts. However, the levels of fibronectin, COL1A1, and TGF-beta1 were significantly reduced. Western blot analysis also confirmed that HDM stimulation activates the NF-kappa B signaling pathway and significantly increases the expression of inflammatory cytokines in bronchial fibroblasts. ELISA results further showed elevated levels of IL-1 IL-6, TNFSF15, CXCL1, CXCL2, CCL2, CCL5, and CCL20 in the cell supernatant. In summary, the uniqueness and innovation of this study lie in the use of primary human bronchial fibroblasts and transcriptomic methods to systematically characterize genome-wide changes. Investigating the inflammatory phenotype of bronchial fibroblasts provides valuable therapeutic insights into asthma-related airway inflammation. You are welcome to cite this study. Thank you all for listening.